And I want to talk a little bit about a set of projects, focusing on one particular project and in some ongoing work. My group's looking for other collaborators, other ideas around this central hypothesis that I'll talk to you about. But my interest is in DNA methylation pattern analysis in perinatal onset diseases. I do not have anything to disclose for this. The specific perinatal disease I'm going to talk about, the one that we're working on actively, is cerebral palsy. It is prevalent in about 1 in 345 children. That's the CDC number, with a 30% higher prevalence in males. It's associated with a static encephalopathy. So there's a problem with the brain that's acquired perinatally, and that's not progressive. That's by definition. But there are progressive peripheral effects, including stiffening of joints, sort of alterations in bone and musculature and movement control. And there's a diagram on the right-hand side that a friend, Steve Cook, who was a surgeon here, drew for us a while ago, kind of showing the components of what's involved with CP. But for us, the real key to this study anyway is that it's perinatal. The children that have it, most of them are born with it. You can acquire CP postnatally, but most children are born with it. But diagnosis is significantly challenging. So for a deficit that is present in a newborn, diagnostic age is around 2, and sort of confident diagnosis often doesn't happen until 5. And that's largely because of the complexity of the disease, the development of movement disorder having to develop to get that solid diagnosis. So we're really trying to figure out ways to identify kids earlier. So the risk factors for prenatal, perinatal causes of CP include things you probably have heard of. Premature infants have a higher incidence of CP, hypoxia, ischemia around birth, placental insufficiency, intrauterine growth restriction, chorioamnionitis and other prenatal infections, perinatal inflammation, sort of as a generic term, and then genetic causes. And the genetic causes of CP have been subject of intense research over the past few years. It turns out, depending on how you think about what the genetic causes are, it's somewhere around 7 to 30% of CP may have an association or linkage with a genetic finding. Whether those are causative or not is unclear. The number of kids that have CP because of a genetic cause is unknown. But by and large, most kids with CP are not genetic. So you can think it's more of an injury model or an exposure model, and sort of an environmental exposure. If you're the fetus, the environment is infected, or there's high levels of cytokines, or there's hypoxia, ischemia associated with your birth. All right, so we're going to switch gears for a second and talk about DNA methylation. So DNA methylation is common. DNA methylation occurs a lot in different parts of typical development. And this list in the second bullet: hypoxic exposure, bacterial infection, inflammation, growth restriction, early life trauma or physiologic stress, all those things that are associated with non-genetic onset of CP are also associated with alterations in DNA methylation. And the paper on the right is looking at preterm birth and sort of the trajectory of epigenome-wide methylation changes. So the other thing that a feature, right? So it's common. DNA methylation changes are common. So differences in DNA methylation are associated with onset, or the conditions that are associated with the onset of CP. And once DNA methylation is changed, those changes are passed on to daughter cells, and they can be sustained quite late in life. So DNA methylation change may be dynamic, so you can put the methyl group on, take it off over a relatively short time frame. Some are stable. And when I'm talking about putting a methyl group on or off, this is a nucleus and then a chromosome and chromatid and DNA all the way down to one of the building blocks of DNA. I'm specifically talking about a methyl group. It's called 5-methylcytosine. So it's a methyl group that's on a cytosine in the context of a CpG. So it's a C followed by another, by a G in the DNA. So typically, although this is sort of a rule of thumb, not a hard rule, methylation indicates repression of gene transcription. That's not always true. Sometimes methylation just means there's a methyl group there, but that's what I'm talking about. Specifically, there are other methylation events that can happen, but we're specifically talking about CPGs. And the way DNA methylation is controlled is at least partially understood. So there's DNA methyltransferases, and I won't go into all of the vagaries and the differences between A, B, and L, but the DNA methyltransferase, DNMT3, will do what's called hemimethylation. So this is the CPG group that I was talking about on the puts one methyl group on. Um, there's a DNMT1, which methylates the other group in a hemimethylated. So that's called maintenance methylation. So you can think when a cell divides and the DNA strands split, the second strand of DNA gets remethylated by DNMT1. There's a process of passive demethylation that occurs, especially when DNMT1 and 3 are low. The methyl groups can just come off. And there's also an enzymatic or active demethylation that happens by a group of enzymes called TET, and they convert the methyl group into hydroxymethyl and other metabolites that eventually then come off. So there's this dynamic state for any particular CPG, right? This cytosine in the CPG group about whether or not it's methylated. And like as I mentioned before, cells and tissues control this process all over the place during development and in pathology. And in CP, this perinatal insult is associated with changes in DNA methylation. All right, so just to close out that idea, you can, if you stimulate DNN, the DNA methyltransferase 3, DNMT3, you can hypermethylate. You get more methylation because you've got more enzyme, as long as the CPGs are exposed, and DNMT and TET expression or the regulation of their complex. So I showed very quickly the nucleus and then the chromosome and then the chromatin. Chromatin structure drives the activity of these enzymes. The levels of certain metabolites, that's supposed to be an alpha, alpha ketoglutarate being one, can have an effect on enzyme activity. There's specific binding partners for these enzymes. So if those binding partners are present or different binding partners, the activity changes. There's post-translational modifications to the enzymes and to their binding partners. And there's a key and rather complex interaction with non-coding RNAs to drive DNA methylation. But you can envision if you kind of turn up the ability to methylate de novo, you can get hypermethylation or methylation at sites that weren't supposed to be methylated to begin with. If you knock down or inhibit DNMT1, maintenance methylation across cell division just doesn't happen, so you can demethylate. And then either inhibition or activation of those TET enzymes could lead either to hypermethylation at a site or hypomethylation. So it's complex, and the sort of mechanisms that control it are complex. But you could reasonably argue that a significant perinatal exposure, and again, we're talking about hypoxia, ischemia, or inflammation, things that are associated with changes in methylation. So if you think about a significant perinatal exposure event, for example, one that's capable of permanently altering the brain in CP, that event would be expected to alter methylated DNA patterns in the affected newborn generally. So CP itself is sort of specific. It's brain neuromotor activity and motor control, but what's precipitating the CP, and this is sort of where we get into the hypothesis that we're driving at. We think the whole fetus is exposed to other tissues which show methylation changes, even if they don't show changes or they're not involved in the pathophysiology. And then I'm going to very quickly go through a paper from a couple years ago to highlight this. This is work from my group in collaboration with Aaron Crougy, who's actually moved on to a company. Adam Marsh is down at UD and is the brains behind the machine learning part of all of this. But the hypothesis from that study was that at the onset of their conditions, the condition individuals with spastic CP had acquired altered DNA methylation patterns in hematopoietic progenitor and stem cells. Those are those cells that make blood cells. And that within this pattern, there's a common, and I'll say stable, the hypothesis is stable DNA methylation signal that's indicative of spastic CP. And just to remind people, this is what we're talking about, that present in the fetus, these myeloid and lymphoid progenitor and stem cells are present, and that the daughter cells from them, whether they're producing other stem cells or they're producing the cells that circulate around in your blood, would pick up the DNA methylation pattern change. That change might be dynamic, so it may only be there in the newborn period, and then say it's methylation, and then the demethylation is corrected, or it may be long term. In fact, there's evidence that these perinatal exposures can end up altering methylation patterns throughout life, basically, because of the effect on the progenitor and stem cell population. So we looked at kids. So we did what's called a case control study. We want to do studies, and we will be doing studies in the next year looking at younger patients. But the first study we did was a case control. So we had kids with CP and kids with non-CP, 16 of each. We collected their peripheral blood cells, and then we did a genome-wide analysis of methylation patterns using a methylation restrictive enzyme approach, a methylation-specific restriction enzyme mediated approach with DNA sequencing. And what we saw, so there's about what we found. This is the target of the enzyme we used. What we found is about 1.47 million CCP-GG sites that were in common across all 32 kids. They were unrelated. So that was our target, 1.47 million. About 62,000 of those had enough variance in them for us to consider in our models. 1.47 million pieces of information is a lot for even a fast computer to deal with. So we use 62,000, figuring at least 10% variance would be necessary to contribute to any model that separates the two. And then we did, this is something like a PCA, if you're familiar with the principal components analysis. It's actually a non-metric multidimensional scaling. It's just a way to look at complex information and whether or not that information allows you to divide the groups cleanly. So what we found is that by NMDS is that the CP group, based on DNA methylation, completely separated from the control population. And then we went in, instead of looking at 62,000 sites, again, that's a lot for computers to work on. We looked at the top 200 sites based on the sort of the statistical difference, and we used false discovery rate corrected p-values to identify that. This is Benjamini-Hochberg, for those who want to know exactly what we did. So we took the top 200. Here's a map. I'm going to zoom in on this in a second. This is called a Circos plot. So as you go around, each of the chromosomes is indicated. And then we just start at the beginning of the chromosome and go across. So any place in these chunks of chromosome, megabyte, megabase, you can tell I work with data a lot. A megabase set of DNA, if it's hypermethylated, it would be red. So that means there's more methylation in that megabase segment in CP than in control. If it's low, then it's green. And I'm not sure you can see these dots. And then these are kind of like little zip codes. There's genes out here. They are not necessarily genes that are affected in their expression. These genes might not even be expressed in the blood cells we're looking at. It's just the location for the top 200 sites as a way for us to track them. So if you zoom in here, you can see this is just chromosomes. These are the smaller chromosomes toward the end, but I use them because you can see a significant amount of hypermethylation in chromosome 18 and hypomethylation in chromosome 19. And it turns out that they're within specific genetic elements, there's up and down sort of hyper and hypomethylation. So the changes in methylation that we saw, that NMDS plot where everything was separated, those are specific to different chromosomal regions. It's not randomly distributed. And I should say the gray here, that's the sites of in the one that weren't statistically significantly different. But at this level of a megabase, there were significant differences in methylation in the CP kids versus the controls. And then we filtered those down. We got the 200 rank sites that was in the Circos plot. And then what we want to know is, do those sites help us tell whether or not someone has CP or not? So we did what's essentially a bootstrap analysis where the computer from those top 200 CPG sites, individual sites, would randomly select a number between 15 and 40. And then we had 16 of each. So it would take 8 CP kids and 8 controls and leave the rest of them. and it would train a model using what's called linear discriminant analysis. That's just a way to tell the difference between the two groups. And it would do this 20 times, just resampling, resampling, resampling. And then as the computer learned, if the sensitivity and specificity that sort of does it call the yeses and nos correctly? So if the sensitivity and the specificity were good across all 20 times that we repeated the set of CPGs across the cases and controls, then we bumped that set up in the model so that we could generate strong models. So after running about 6.8 million for this paper, LDAs, there were 252 different models based on less than 40 CPG sites that were nearly perfect. And we say nearly perfect, they were 100% accurate, which strongly indicates that there's a signal, a potentially diagnostic signal in the methylation patterns for those kids. So the differences in the participants with spastic CP were readily distinguished. It was actually much stronger signal than I was expecting. I think the folks that work on methylation for a living weren't surprised, but I was very surprised at how strong the signal was. The distinguishing patterns remained when we took those 252 models that we generated and we applied them now to kids who were about 4 years old. The accuracy was very high. In fact, the accuracy in the computer diagnosis was about the same as clinicians, clinical diagnosis for that age group. So the results suggested that a blood test may be possible on a small panel of less than 40 CPGs for spastic CP. There's clear limitations. These are older kids. It only looked at binary, so we looked at CP versus non-CP. That's not what a labor and delivery room looks like. And we didn't really look at the origins or the consequences of DNA methylation, just the signal from DNA methylation. All right, and moving forward, it turns out we know it's less than 40. This is Jonathan. He's actually adding in complexity. So we're looking at kids with concurrent epilepsy. This is a 3D. I kind of like this. He doesn't, but I kind of like it. This is a 3D representation of methylation patterns in a principal components analysis for kids with CP epilepsy or control. And actually, it's the number of CPG sites that it looks like we will need is significantly lower than 40. In fact, his latest work shows that maybe 6. So that's fantastic because looking at 6 sites, you could do that literally in the clinic or as either a point of care or a very quick turnaround test. There's a lot of work that needs to be done for us to prove that that works, but that number of CPGs we might need is extremely low. And I do want to mention we are also looking in muscle for these distinguishing patterns. So this is Karen Robinson. So she was the lead author on a recent paper looking at differences in skeletal muscle satellite cells. So for the blood, we're going after biomarkers or signals for diagnostics. In the skeletal muscle satellite cells, which are part of the target, one of the target tissues in this disease, we're going after potential pathways. There were a lot of people that work on CP project with us. So I do want to point out Adam Marsh, who originated the algorithms and the pipelines that we used for that paper and that we're using going forward, and Aaron Crougy, who helped gestate and develop this idea from the beginning. And then the CP group, especially Freeman Miller, Jason Howard, and Wade Schrader, amongst a number of other people from the lab. And we did have the funding for this project, the trajectory of it, not just for the one study that I talked about, has come from a number of different places, including NICHD, the Swank Foundation, the Delaware Cat Group, and the AACPDM. So I want to thank all of them and thank the patients and the clinicians who help us with the study. And for you guys, I want to thank you for your attention. But this idea, so go back to the title, that these perinatal onset diseases, what we're finding is, and again, I'm as surprised as anyone that these signals are there, that looking at epigenetic signals may give us a handle on what happened at some level, what happened to the point so that if you have an infant that's at risk for CP, the blood test may put that risk into a finer focus. And for CP, that's the perinatal disease we're looking at. There's a lot of kids that are at high risk. They hit a lot of those risk categories that come to the NICU or come to neurology that don't develop CP. In fact, the sort of overall incidence of CP in at-risk patients is much lower than you would think. So those risk factors are helpful, but they're not really diagnostic. since in some cases, depending on whether it's preterm or when the baby was born and what the risk factors actually are, it's less than 20% of the kids considered at high risk actually develop CP going down the road. So the addition of this simple blood test to say this child really is high risk and this child is lower risk would help enormously in study design, in getting care assigned to those patients, and in helping parents and families and clinicians understand the trajectory for those patients going forward. And we think that that idea is generalizable to a number of other perinatal conditions. So thank you all very much. I know I talked for longer than I tell you guys to talk, but I haven't been able to present my science in a while. And if anyone's interested in chatting, I would love to hear from potential collaborators if anyone has ideas. But thank you for your time.